Thank you. Sam had asked me to speak about pesticides, the group of contaminants in which I have spent much of the last decades concentrating on. Speak about pesticides around the Delta. The data doesn't exist to speak about pesticides strictly in the context of interior Delta flows. But I think I can give you a flavor for some of the pesticide stresses that these same fish species, the smelt, the salmon, might encounter as they move about the Delta. So the levels of pesticides, I think generally speaking it's safe to say, are not at a concentration where we would expect acute lethality in the fish. They're 1 to 2 orders of magnitude too low to simply kill fish species outright. Sublethal effects, I don't think I can unequivocally dismiss, however. They're certainly a consideration. But the concentrations we see are high enough to be acutely toxic, acutely lethal to a number of invertebrate species upon which the fish depend. And so much of my work has focused on that aspect, effect on the fish, but indirectly via loss of some important prey species due to pesticides. So it's the invertebrates that I'll be focusing on today. First, to give you I guess a sense of why addressing, what are the pesticide impacts on fish or the invertebrate prey in the Delta is such a thorny problem. I've listed here some data compiled by Queevil and Hladik showing the pesticide use in the San Francisco Bay watershed. So this data includes more than the Delta, but the Delta data would be very similar. This is throughout the watershed showing how many herbicides, fungicides, insecticides, and others are used. And also subdivided into those that have any monitoring data, and I don't mean regularly, but ever, versus those that have never been monitored. If you put that all together, we've got herbicides, something on the order of about 60 herbicides used in the watershed. Only a little more than half have ever been monitored. So a little bit less than half have no monitoring data whatsoever. We have no idea what the concentrations are in the environment. Fungicides, 32 of them, nearly all no monitoring data whatsoever. Insecticides, about 50, about 40% of which have never been monitored. And then there's a few in the other category. The yellow text moved from when I saw it on the slide earlier and moved up to where it's unreadable. But what that yellow text says is that there's a total of 160 pesticides used in the San Francisco Bay watershed. And 55% of them have never been tested for in the environment. So again, when you ask me to speak on pesticide impacts, again, I've got a very limited data set to work with simply because we're not measuring over half the compounds that are being used. To make matters worse, I've listed 2 points there. Environmental degradates, which are sometimes more toxic than the parent compounds, are rarely monitored. So even if we're monitoring for the particular herbicide or a particular insecticide that is being applied, we're probably not monitoring for the substances that degrades into, which are sometimes more toxic than the substance applied. So that 160 number quickly inflates to several fold if you consider potentially toxic degradates. And also, it may come as a surprise to know that pesticides have been approved for use by regulators even when the best analytical labs are not able to measure environmental concentrations exceeding toxic thresholds. So a pesticide could be in use, it could be in the environment at concentrations that are toxic to aquatic life, but the labs are still unable to measure it. You might think that might dissuade approval and use, but you'd be wrong. It does not, unfortunately. So those are some of the challenges we face. Pesticide use is also a moving target, and what I've done here is graph non-agricultural use, so this is mostly urban use, around residences largely, in California of selected insecticides. So I've got data from 2000 to 2011, which is the most recent available, showing how much in kilograms of these various insecticides have been used in non-agricultural settings in California. You can see the one that has really dropped off are the organophosphates, diazinon and chlorpyrifos, going from almost 500,000 pounds in 2000 to basically none currently. That is actually I guess, one of the more important success stories when it comes to water quality over the past few decades. Back in the 1980s, 1990s, lengthy reaches of the Sacramento and especially the San Joaquin River would go toxic every winter or following a winter storm. Diazinon typically was the responsible agent. Concentrations would spike after the rains, and it would be toxic to sensitive invertebrates over 30, 40, 50 miles of those rivers. Again, the Sacramento and the San Joaquin both had those effects. The San Joaquin concentrations tended to be much higher. We don't see that today any longer. Two reasons. One, because the non-agriculture use has dropped to zero. EPA removed them from the marketplace, not because of water quality issues, because of human health concerns, but it had the unintended benefit of solving the water quality problem. So they have not been purchasable for urban uses since early 2000s, and that's why that graph goes down to zero. On the agricultural side, they're still used, but not near the quantities they used to be. That's as a result in part of grower education, teaching growers better ways to apply to reduce runoff. In part, unfortunately, it's a switch to pyrethroids. As the organophosphates came under regulatory scrutiny, some growers simply switched to pyrethroids. The pyrethroid data, you can see, it was about 200,000 kilograms at the beginning of I guess this century, 2000. Climbed up to about 400,000 and then dropped, which I'm guessing is due to economic conditions. As the recession got worse, no houses not being built with the frequency they used to. That means less termite treatment required. You see the pyrethroid use come down and is now starting to climb as we move out of the recession. And then fipronil is an emerging compound, hardly any use at all 10 years ago, but its use is climbing, and you can see the basic shape of the curve basically mirrors that of the pyrethroids. I'm going to today talk about those two categories, pyrethroids and fipronil. First, to give you a sense of the species that we normally use for testing water quality. Sort of the standard species for many years has been Cereodaphnia dubia, which is that one to the left, right there. Small crustacean, about the size of head of a pin, maybe. And I've shown the LC50 for bifenthrin and fipronil for that species. If you're not familiar with LC50, that's lethal concentration to 50% of the test organisms. So that's the concentration that if you added to a beaker would kill half of your Cereodaphnia, or whatever the species may happen to be. It's the concentration that kills an average Cereodaphnia or an average Hyalella or an average Chironomus. So you can see the average the LC50 for Cereodaphnia is 50 nanograms per liter. On the left, you'll see the highest concentration we've observed in creeks, urban creeks, is 106 nanograms per liter. So if you're testing with Cereodaphnia, you're only going to see toxicity in a few of the very most contaminated water bodies. For the most part, you won't see toxicity. Hyalella is a species we largely use when we're interested in pyrethroids, right there, that's Hyalella. Because it's got a EC50, that means the concentration causes paralysis. In this case, it doesn't really kill them, but it paralyzes them. And I think in the real world, a paralyzed invertebrate is basically a dead invertebrate. You can see Hyalella is far more sensitive to the pyrethroids. Bifenthrin, in this case, is the pyrethroid the data represents. At only 3 nanograms per liter. That's 3 parts per trillion. It's a phenomenally low level, but yet it is toxic to Hyalella. And then Chironomus, which is that red, wormy-looking thing all the way to the right, very insensitive to pyrethroids with a EC50 of greater 253. Fipronil, Cereodaphnia is a horrible species to test for fipronil. The highest we ever have seen in the environment is 49, and you don't kill Cereodaphnia to get to 17,500. You could sample for the rest of your life and never find a toxic sample if you're using Cereodaphnia. And Cereodaphnia is what the agencies have relied on for many decades because it happens to be very sensitive to organophosphates, and they historically have been the major concern. Hyalella, not much better. Chironomus, however, very sensitive to fipronil with EC50s below that of the concentrations we see in the environment. So the point is, when I say water sample is toxic, it depends on what species you're using to define toxicity. A water sample that's with pyrethroids is very likely to be toxic to Hyalella, probably not to Cereodaphnia, and certainly not to Chironomus. A water sample with fipronil is going to be toxic to Chironomus, never to Ceredaphnia. Okay. The 4 data sets I want to present today, and I'm going to have to present just a quick overview of them. I don't have time to get into details. Pyrethroids in the American River. Technically, that's not in the delta, but just barely outside of it by a couple miles. Pyrethroids in the American River. Pyrethroids in Cache Slough. We've heard about Cache Slough a number of times in the presentations over the past couple days. It's in the northwest delta, a very important habitat for spawning delta smelt. Fipronil in urban creeks. And then an effective contaminants that we often don't think of and have generally gone unrecognized, but we now have data to indicate is quite significant. Evolutionary adaptation to pesticide exposure. I'll show you some data indicating that we've changed the genetics of a organism in California due to pesticide exposure. So first, pyrethroids in the American River. Here's a photo of the American River just below the Folsom Dam. The American River is considered one of the cleaner rivers around here. There's a hatchery on it. It's clean enough that there's a drinking water intake on it. It's quite a pretty river, and again considered one of the cleaner. However, if you look at the flow, you find that in a substantial rain event, a large portion of the American River flow, by the time you get to Sacramento, is in urban runoff. So this is a January 2010 storm, produced 3.5 inches over 6 days. As the American River left Folsom Dam, which would be all the way over here on the right, it was carrying about 1,500 CFS. Each of these step-ups is stormwater entering through a creek, a pump station, a concrete channel, whatever. If you add the flow of all those entries to that base 1,500 CFS, by the time you get to the confluence of the American River with Sacramento River, basically downtown Sacramento, the American River is now carrying 2,600 CFS. So the difference between the 1,500 when it left the dam and the 2,600 when it gets to the Sacramento is runoff. So that urban runoff is 40% of the river flow. When we look at the American River, we generally don't appreciate that. But because of that 30-mile stretch is so urbanized, much of the flow is, again, urban runoff. And what does that urban runoff carry? This is showing inputs of stormwater runoff to the American River, represented by the arrows, and the number represents the bifenthrin concentration presented as the number of times over the Hyalella EC50. In other words, if 3 nanograms per liter was the paralyzing concentration, the EC50 for Hyalella, and the stormwater runoff contained 9, then I would say that you see 3 here on the figure, 3 times the EC50. So we've got this first creek just below Folsom Lake, putting in, depending on when we measured it, anywhere from 4 to 13 times the bifenthrin concentration that would be sufficient to paralyze Hyalella. Then we've got 2 creeks with relatively low numbers. Then we've got another input of bifenthrin with 4 to 9 times EC50, 8 to 10 times EC50, 13 to 32 times EC50, and on down the river, all with quite large numbers. The black arrows are inputs that we didn't sample, so I don't have the bifenthrin data for, but you can, I think, safely assume that the numbers are going to be just as large as the ones I do have data for. Bottom line is urban runoff is putting in bifenthrin, a pyrethroid, at about 8 times the concentration on average that paralyzes Hyalella. So if 40% of the river by the time it gets to Sacramento is urban runoff, and that urban runoff contains 8 times the paralyzing concentration of bifenthrin, no surprise that the American River is toxic due to bifenthrin by the time it gets to the lower reaches near Sacramento. Here I've plotted the observed concentrations of bifenthrin measured in those last few miles of the river, in basically downtown Sacramento. That's these numbers here. And this other column are EC50s of various invertebrate species. Here's Hyalella down at 3. You can see the American River routinely after storms has bifenthrin concentrations that exceeds the concentrations that would be paralyzing to Hyalella. basically Hilo and that lower reach of the river are toast after a major rain event. The next point up is Hydropsyche, a caddisfly, with an EC50 of around 12. 14 is the highest we've seen in the river. We get quite a few, maybe around 6 or 8. Again, 12 is what it takes to paralyze this particular caddisfly. The significance to salmon, and for that matter steelhead, comes into the fact, based on stomach content analyses of both Chinook and steelhead done in the American River upstream at this point, Hydropsyche is the most important prey species to those fish on a biomass basis. It makes up the majority of the biomass in the gut contents. So I can't tell you that the concentrations of bifenthrin we see in the American River are going to deplete the available Hydropsyche prey for salmon, but if not, it's pretty close. You can see that Hydropsyche's sensitivity is right at the upper edge of what we're seeing in the river when it comes to bifenthrin concentrations. Cache slough, again, a very important habitat for spawning of delta smelt. We routinely find toxicity in cache slough following winter rains. The area of concern is Eulatus Creek. That's that red line coming in from the left, and then where it bends up to the right a little bit, that's where it meets cache slough. And then this area of cache slough from that juncture down to the mouth of Lindsay Slough, and for a few miles up Lindsay Slough, routinely go toxic to Hyalella after winter rains. I've sort of summarized the work here on the left. Eulatus Creek carries Vacaville urban pesticides supplemented with agricultural inputs 13 miles to cache slough. I mean, look at the photo. It's clearly a lot of agricultural land around cache slough. I think before we did this work, I and probably most people would be shocked to hear we had an urban runoff problem. But in fact, I think we do. It's Vacaville urban runoff being carried 13 miles with its pyrethroids still intact and going into cache slough. 2/3 of the samples within that cache slough, Lindsay Slough area, the red circled portion on the map, following 4 winter storms, were acutely toxic to Hyalella. So pyrethroids are entering at toxic levels regularly. The substances responsible were the pyrethroids bifenthrin and on one occasion cyhalothrin. Bifenthrin was a combination of urban and agricultural input. The cyhalothrin was agricultural input from one particular drain that we had identified. The highest bifenthrin concentration observed, 7 parts per trillion, may be at the LC50 for the copepod Eurytemera finis, which is an important delta smelt prey species. Copepods obviously are important for smelt. We were interested in whether these concentrations would be toxic to those key prey. The test was done at 20 degrees centigrade. The LC50 at 20 is up in the teens, but pyrethroids are unusual and they get more toxic as temperature goes down. And certainly cache slough in the winter is not at 20, it's more like 8, 9, 10. If you factor in the lower temperatures in situ, then that LC50 for the copepods that's up in the mid-teens probably drops somewhere very close to the highest bifenthrin concentration we observed, of somewhere around 5 to 10. So those concentrations are certainly toxic to Hyalella, and they may be toxic to, again, an important delta smelt prey species. And just so you don't think I've cherry-picked the data here or picked on Sacramento's runoff or Vacaville's runoff, I've shown all the cities in which we've collected urban runoff and the concentration of bifenthrin in those cities. The red line represents the concentration of bifenthrin that is the EC50 to Hyalella. Anything more than that will paralyze Hyalella. And again, paralysis, I think, is basically closely equivalent to death for an animal in the wild. Novato, Oakland, Fremont, Vacaville, Stockton, Elk Grove, Sacramento, Carmichael, Rancho Cordova, Fair Oaks, Nimbus, Folsom, Roseville, they're all way over that red line. You'd be hard-pressed to find any urban runoff in Northern California that does not have bifenthrin that's concentrations that are toxic. So basically, in any portion of the delta where you've got substantial amounts of urban runoff entering, you have the potential for toxicity to sensitive aquatic species, particularly the invertebrates. Now, Fipronil, again, a relatively new pesticide, and many of you have probably used Fipronil. If you have a dog or a cat and you've used Frontline, you've used Fipronil. Now, I'm not blaming Fido for contaminating urban creeks here. Fipronil has other uses around homes for controls of ants and termites. There is no agricultural use permitted in California, so it's entirely an urban signal. And we have started sampling it recently. The problem with Fipronil is it quickly degrades to any of a number of compounds, the sulfone, the sulfide, the desulfonil, the amide. Many of those are about 3 times more toxic than the parent compound. So no longer are we tracking just one compound, we have to track 5. So that certainly complicates things. We have sampled a number of urban creeks, and I'm not going to go through them, but you can see we've got pretty good coverage throughout California there, about 20 creeks in many cities. I've shown, sorry, but the computer messed up the slide. It wasn't like this when I prepared it. What that says on the bottom, these columns on the right, in other words, that one for Fipronil, that one for the sulfide, and that one for the sulfone, show the concentrations in the urban creek waters. All right, so we've got, 24 samples maybe. Each of those dots represents a sample. The points on the left side, that for Fipronil, that for the sulfide, that for the sulfone, represent the EC50s to a variety of invertebrates. And you can see that the actual concentrations in the creeks are at or above the EC50s for several benthic invertebrates. In that case, there's Fipronil. The highest concentration we see is certainly well above that most sensitive species, that's Chironomus. It's even basically at the EC50 for the next most sensitive species, which is a baited mayfly. If you've worked with salmon diets, you know baitids are quite important in salmon diets. The sulfide is not as much of a problem. It's certainly a problem for Chironomus. The other ones are higher. The sulfone, we've got many creek samples that are way above the concentration that's toxic to Chironomus, and approaching the concentration of, in that case, it's a caddisfly, and I don't recall what that one is. But the point is, the concentration of Fipronil and its degradants in the creek are routinely above the concentrations that are important to Chironomus, paralyzing to Chironomus, and at or above the concentration to often 1 or 2 other macroinvertebrate species. Just briefly, I'm going to skip that just for the sake of time. Okay. Now, as far as the genetic effect that I alluded to earlier, there's Hyalella. We and others have found locations from which we take sediment or water, bring it back to the lab, put the Hyalella that we've been rearing in the lab for many years into those samples, and they all die. But we find a thriving population of Hyalella in the same creek from which we took the sample. All right? So we like to think toxicity to our cultured Hyalella is predictive of the environment, but again, the wild Hyalella are doing obviously just fine, thank you. And so this was a bit of a obviously concern and a puzzle. So we looked into it in greater depth. Here is the cyfluthrin LC50. So this is the concentration of cyfluthrin, another pyrethroid that would kill Hyalella. For our lab cultures, one from Virginia, one from Illinois, mine from Berkeley, you're looking at anywhere around 1 to 5 parts per trillion cyfluthrin is the concentration that kills them. We looked in 2 fairly remote sites in Northern California. These are water bodies with minimal agriculture, no urban input. These guys probably haven't seen any pyrethroids to speak of. And their LC50s are just like our lab cultures in that 1 to 5 sort of range. However, you look at creeks containing Hyalella in Northern California, and these are creeks that are either going through urban environments or agricultural environments, and look at the LC50s. The best is down here at 10. We've got some here greater than 691. In other words, at 691, we still couldn't kill 50% of the animals. So it's LC50 is something greater than that because we just can't kill them. We can't determine an LC50. So we've got these populations in creeks that have been for many years dosed with pyrethroids, unfortunately due to urban runoff, that can handle hundreds, if not almost 1,000 nanograms per liter, far more than an average might not be the right word, a naive hyalella that hasn't seen pyrethroids before. I find this fascinating. We're seeing at least a 2, maybe even 3 order of magnitude difference in sensitivity in hyalella to pyrethroids. How do they do it? Well, as we got into the genetics of it, we were able to answer that. Pyrethroids act on what's called the voltage-gated sodium channel. There's a protein about 200 amino acids long that forms an opening in the neuron for sodium to go in and out. And the neuron controls the flux of that sodium by opening or closing that port. What pyrethroids do is they bind to that protein and lock it in the open position. So the animal cannot control sodium flux across the neuron, and the neuron just fires and fires and fires and fires, and the animal dies of exhaustion. The dark, the black spots here that you see are where mutations have been reported that provide resistance to pyrethroids. Pyrethroids obviously are being used for pest species, so agricultural pests, mosquitoes, and in many cases pest species have developed a resistance to pyrethroids, and those black spots indicate where on that protein that the change occurred that provided this resistance. So we sequenced the DNA of these hyalella. Here is a lab-cultured hyalella, in other words, hasn't seen pyrethroids before. And here's a stretch of the amino acids of that 2,000 amino acid long protein. Here's a stretch of about a dozen amino acids. And here's the DNA that codes for those amino acids. So its genotype is CTC, and you get a leucine when you transcribe that. Then it's got an AAC, and you get an asparagine, etc. What these resistant hyalella have done is here at position 918, where you have ATG, which should give you a methionine, that adenine has mutated to a thymine. So you get a leucine in that position instead of a methionine. Or they would have one or the other of these mutations, but not both. Over here, position 925, that CTC, that C has mutated, that cytosine has mutated to an adenine, so you get an isoleucine instead of a leucine. Either one of those mutations changes that voltage-gated sodium channel protein, preventing the pyrethroid from binding to it, and therefore the function of that sodium channel is not impaired. So what these hyalella have done is the very same thing that the mosquitoes and the other agricultural pest species have done, is the few resistant individuals in a population that have these mutations just by sheer luck have been selected for. The sensitive species have been eliminated, and we're finding populations that have become, over time, resistant to pyrethroids. That's why we're seeing such dramatic differences in the LC50s. Here's one site. This is a creek in Roseville. And again, I'm sorry that the software screwed up the text. It wasn't like that when I saw it. Here's showing the sensitivity of hyalella. This is how many, the percent survival going from 100 down to 0 as you increase the cyperthrin concentration. And here in 2010, that's the left curve, you get to about, I think that's about 10. Yeah, about 10 parts per trillion, they start dying. The LC50 is 11. By the time you get to 50 parts per trillion, they're all dead. 3 years later, we went back to the very same site, and now the population looks like this. That 50 parts per trillion that killed everything doesn't bother them at all. The LC50 has shifted from 11 up to 96, and you can go all the way to, in fact, to 1,000, and you still have 10% of the animals still there. That population has become more resistant just in the 3 years between these two intervening sampling periods. It's like evolution right before our very eyes, which I just find really fascinating. So, I think we have again, changed the genotype of Hyalella azteca in Northern California, and I'd even bet nationwide, though I don't have the data to prove it, because of this pesticide exposure. How much more of this is going on? Up until a few years ago, we couldn't have even done this because we didn't have the genetic tools to do it. Now we can sequence the DNA and with relative ease. Easy for me to say relative because I don't have to do it. One of the collaborators do. But anyway, we can sequence the DNA, figure out how this resistance emerged. So we're seeing this in Hyalella. People were unaware of it until we published on this just a few months ago. How many other species are out there this is happening to? Whether it's Hyalella, whether it's fish species, where pesticide exposure or any other contaminants for that matter are changing the genetics of the population such that they're still living, and we say, oh, this water body must be fine. It's got Hyalella, can't have pyrethroid problems. I mean, that would have been the old interpretation. If you saw Hyalella in a creek, well, then it didn't have pyrethroid problems in that creek. We know now that that's not a fair assumption. How much more of that is going on that we're oblivious to? We're making inferences on what the habitat is like based on what species are living there. How do we know that genetically those species have not been altered? Just something to think about. So I've whizzed through this pretty quick. I've listed 4 publications. All the data I presented, well, 90% of the data I presented, has been published in the past couple years. And if you're interested, there's the citations to get further details. All right, thank you.
Somebody has one quick burning question. We can probably fit it in. Okay, hearing none. Next speaker is going to be the discharger perspectives and Tim Musson and Tom Grovo.